Good evening, everyone. Myself, Dr. Monika. I am from Department of Sciences, Trinity College, Jalandhar. Today, I am going to discuss about the topic biotechnology and its applications. First, we will discuss what is biotechnology and its complete process. Biotechnology is the use of living organisms or processes to make products useful for the mankind. The term biotechnology was first used by Carole Aricki in 1919. In traditional biotechnology, which include production of bread, beer, wine with the help of fermentation process. In this process, in the first example, we can see that for making the bread, we used yeast species Saccharomyces cerevisiae, which is mixed with sugar, flour, and warm water to make the bread. It is a single cell fungus. It is able to ferment sugar and to produce alcohol and CO2 in the process. And the second example is yogurt. Yogurt is a fermented milk product in which we convert milk into a lactic acid. Bacterial species used in this process are Streptococcus thermophilus and Lactobacillus vulgarius. In modern biotechnology, which make use of the advancement in science such as genetic engineering, recombinant DNA technology with great effects. In this technology, we can produce artificial DNA through the combination of different genetic material from different sources. This process, popularly known as genetic engineering or genetic cloning. Now, let us discuss about the process of recombinant DNA technology in detail. In the first step, we isolate the genetic material. It is the initial step in recombinant DNA technology to isolate the desired DNA in its pure form, which is free from other macromolecules. In the second step, we cut the gene at the recognition sites. For this, restriction enzymes play a major role in determining the location at which desired gene is inserted into the vector genome. And these reactions are called restriction enzyme digestions. In the third step, we amplify the gene copies through polymerase chain reaction. It is a process to amplify single copy of DNA into thousands to millions of copies once we insert the proper gene of interest. After that, we ligate our cut fragment of DNA plus vector DNA with the help of DNA ligase enzyme. In the last step, we insert our recombinant DNA into the host. This process is also termed as transformation. Once the recombinant DNA is inserted into the host cell, it gets multiplied and expressed in the form of manufactured protein. In this diagram, we can see that first we treat our foreign DNA plus plasmid with restriction enzyme, and after that we ligate both the fragment with the help of DNA ligase enzyme. After that, in the second step, we introduced our recombinant plasmid into the cultured plant cells. At the end, we can regenerate our new plant from these culture cells with new trait. Now, let us discuss about the applications of biotechnology in agriculture. With the help of green revolution, we increased our production of crops by using pesticide, fertilizers, but excessive use of these chemicals polluted our soil, our environment, as well as our health. So, with the help of biotechnology, use of these pesticides, fertilizers are greatly reduced. With the help of genetic engineering and recombinant DNA technology, we can produce genetically engineered crops by inserting gene of interest into these species. There are many advantages of these genetically modified plants. Let us discuss them one by one. The first major advantage is tolerant to abiotic stress. And the second one is pest resistance crops. By producing these pest resistance crops, we can reduce our dependency on harmful chemicals. And the third one is reduced post-harvest losses to enhance the nutritional value of the food, like production of golden rice, which is rich in vitamin A, and to increase the ability of mineral uses by the plants. Let us discuss about various examples of genetically modified plants. First, we will discuss about Bt cotton. There are some strains of Bacillus thuringiensis which produce a protein known as Bt toxin, which is highly toxic to the insect pests like coleopterans, lepidopterans, and dipterans. These are those insect pests which are harmful to our crops. Bt toxin is a protein which is present in Bacillus thuringiensis in inactive state. But when insect ingest this inactive toxin, it get converted into active toxin due to the alkaline pH of the gut. The toxin bind to the surface of the midgut epithelial cell and create pores. After that, it causes cell swelling and cell lysis, which leads to death of the insect. You can see in this diagram, after ingestion of this Bt toxin, it gets activated in the gut of that insect and lead to the death of the insect. So, by genetically modifying our Bt cotton crop, we reduce the uses of pesticide as well as fertilizers. Second example is of flower saver tomato. It is a genetically modified tomato, first commercially grown, genetically engineered food to be granted a license for human consumption. It was developed by the Californian company Colgene in the 1980s. In this diagram, we can see the difference between the traditional tomato as well as in the flower saver tomato. Traditional tomato जो था उसको क्या करना पड़ता था हमें, ग्रीन जब वो मतलब कि वाइन के साथ अटैच होता था, ग्रीन होता था, फर्म होता था, उसी टाइम उसको मतलब कि हमें लाइक प्रक करना पड़ता था, लेकिन और उसके बाद उसको स्प्रे किया जाता था विद इथाइलिन टू इंड्यूस द राइपनिंग. लेकिन क्या होता था कि जैसे ये सुपरमार्केट में जाता था तो इसकी जो फ्लेवर था वो काफी रिड्यूस हो जाता था. लेकिन ऑन द अदर साइड विच इज फ्लेवर सेवर टोमेटो, जेनेटिकली मॉडिफाइड टोमेटो, इन दिस बट वी डिड वी जस्ट मतलब कि इसको पहले नहीं हम तोड़ते हैं, पूरा राइप किया जाता है वाइन के ऊपर ही, फुल फ्लेवर डेवलप होता है इसमें. देन आफ्टर दैट हम इसे हार्वेस्ट किया जाता है और इसका फ्लेवर बिल्कुल मेंटेन रहता है जब ये सुपरमार्केट में पहुंचता है. और जो जीन रिस्पॉन्सिबल था जिसको हमने ब्लॉक किया, जो मतलब कि राइपनिंग करता था इस टोमेटो की, वो था पोलीग्लैक्टुरोनेज. इसकी जो प्रोडक्शन थी उसको हमने ब्लॉक कर दिया, क्योंकि ये ही वो एंजाइम था जो रिस्पॉन्सिबल था फॉर द डिग्रेडेशन ऑफ पैक्टिन एंड सॉफ्टनिंग ऑफ द फ्रूट. Or with the help of genetic engineering, this transgenic variety of tomato can stay fresh and retain flavor for a longer duration. And the third example is the production of genetically modified rice, also known as golden rice. In vitamin A deficiency, which is common in African countries, Southeast Asian countries, 50% of the countries globally mainly affected by this vitamin A deficiency, or it may cause night blindness, increases the risk of maternal mortality in pregnant women. In this picture, we can see golden rice, which is a genetically modified rice, and it is rich in beta carotene. The developer of this golden rice projects were Swiss Federal Institute of Technology and University of Freiburg, Germany. Both work on the development of this golden rice, and its main developers are Ingo and Peter. Golden rice, we create golden rice by transforming it with 2 beta carotene biosynthesis gene. We introduced phytoin synthase extracted from daffodil and carotene desaturase obtained from the soil bacteria. So that's all about its application in agriculture. Thanks for your patient listening.